Hi everyone, and welcome to Lab 2. The topic of this lab is aseptic transfer technique. Some of these words may be unfamiliar to you, so we're going to talk about what they mean and define them before we start talking about how the lab is executed. Aseptic technique is defined as the practices that are required to prevent cross-contamination of microorganisms in the transfer of pathogens when working in a lab or medical setting. Basically, it's all of the things that we do to allow ourselves to remain protected and to prevent our specimens from becoming contaminated with unwanted microorganisms. In this lab, the goal is to practice aseptic technique while executing subculturing of the bacterium known as Serratia marcescens into several new types of growth media. So at the same time as learning about how to perform aseptic technique, you will also be learning about the tools that are used to transfer bacteria from one medium to another, as well as about the different types of growth medium that we will be using throughout the semester. Several important terms that you're going to hear over and over again throughout the semester are inoculation and inoculum. Inoculation describes the act of introducing a microorganism into a new environment, and the inoculum is the subject of the inoculation. It is the sample of microorganisms that is being picked up and transferred into the new environment. There are two main inoculation tools that are used to transfer microbial samples from one medium to another. The loop is the one that you see pictured on the top in blue, and this is used when transferring culture to a solid plate medium or into a liquid medium. The needle is used when a transferred culture needs to penetrate a solid media. It's also used for the Agar slant, which is one of the types of media that you'll be introduced to in this lab. The four main types of media that we're going to be looking at throughout the semester are the broth tube, the Agar slant, the Agar deep tube, and the Agar plate. In this lab, however, we are only going to be looking at tube media. Agar plates are something we'll be introduced to a little bit later. The difference between a broth tube and the Agar slant and Agar deep tube is that the broth tube is liquid, while these other two tubes have a solid medium. So let's take a look at these different types of media one by one. The broth tube has a liquid nutrient media, meaning that it is mainly composed of water, but with nutrients mixed in that are required for microorganisms to grow. There is no agar in the tube, and agar is the solidifying agent that causes the nutrient media in the other types of tubes to be solid rather than liquid. The microbes in a broth tube, therefore, are able to grow dispersed throughout the liquid because the medium is not solid. The Agar deep tube, on the other hand, contains the same nutrient broth that is found in a regular broth tube, but it has been supplemented with a compound, agar, that causes it to become solid. Agar is much like gelatin in that when it is added to a liquid substance and then heated up, upon cooling, it will cause that liquid to become solid. The goal of the deep tube is to see where bacteria grow and the pattern of their growth along a straight line stab that penetrates into the media using the needle. So deep tubes are never inoculated using a loop. Instead, you stab inoculate them by driving the needle along a straight line about 3/4 of the way to the bottom of the tube and then back out. The Agar slant tube is similar to the Agar deep tube. It is composed of solid media. However, this media, when it is heated and cooled, is left to cool and solidify at an angle. And so therefore, unlike the deep tube, this media is angled at a slant. The benefit of the slant tube is that the surface area of the slant is greater than in the deep tube. And so this is better for observing the growth pattern of an inoculum that can be spread out over the surface of the slant. And this is how you inoculate it. You use the needle to take a sample of bacteria and spread it back and forth over the surface of the slant. So this slide right here summarizes the different inoculation techniques that should be used when transferring to and from different types of stock cultures. In the lab, as we said, you are going to be transferring Serratia marcescens from 2 different stock culture forms into each of the 3 types of tubes that we've just discussed. You'll be presented with a broth stock culture with Serratia marcescens growing in it, and a slant stock culture with Serratia marcescens growing in it. The goal of the lab is then to take each of these and transfer the broth to a new broth, a new deep, and a new slant, and to take the slant and transfer it to a new broth, a new deep, and a new slant. Then, after incubation, the results of the new growth can be observed. So now let's take a look at what this procedure actually looks like in the lab. All right, everyone, let's take a look at what we have set up here in terms of tools. We have a Bunsen burner, which is going to be used to sterilize our inoculation tools, which includes the loop, as well as the needle. Now, which inoculation tool you use is going to depend upon the form of media that you are transferring into. We have here 3 different forms of media. We have an Agar slant. As you can see, it is full of solid media that is on a slant. We also have a liquid broth tube. And then finally we have an Agar deep tube. Solid media, not on a slant. For our stock cultures, we have 2 tubes of Serratia marcescens. This one is a stock culture grown on a slant. This one is a stock culture grown in a broth tube. We are going to be transferring each of these into one of the 3 different types of media. The inoculation tool that you use depends upon which type of media you are transferring into. If you are transferring into a fresh broth tube, you want to use the loop. If you are transferring to a fresh slant or fresh deep tube, you want to use the needle. So let's get started. I am going to begin with our Serratia marcescens slant stock. And before we begin, we want to make sure that we sterilize the instrument that we're using for the inoculation. My first inoculation is going to be Serratia marcescens from the slant to a broth. Because it's a broth tube, I want to use the loop. So I'm going to take the loop and I'm going to heat it in the flame of the Bunsen burner until it gets red hot. Then I remove the loop and I allow it to sit and cool. You don't want to wave the loop around to facilitate its cooling because this can actually recontaminate the loop with microbes that are airborne. Once the loop has cooled, I am going to take my stock tube and I'm going to use my pinky finger to remove the lid. And this way, by not setting the lid down, we don't contaminate any of our surfaces. I take the loop, I go into the slant, and I pick up a small amount of inoculum from the surface. Then I recap my stock tube. Next I have my broth here. To inoculate the broth, I remove the lid once again. I plunge my loop into the broth and I twirl it in order to mix. This dislodges the microbes that I have picked up from the surface of the agar slant. Now that the broth is inoculated, I'm going to set it over here in this rack, and I need to reflame my loop to kill any remaining microbes that are left behind. Once again, when the loop is red hot, we remove it and allow it to cool. Next, we're going to take that same Agar Slant stock tube and we're going to inoculate our Agar Slant fresh tube. This time, we're going to be using the needle for this job. So I want to flame the tip of the needle until it's red hot. And then allow it to cool once again. In the same fashion as before, I pick up my stock tube, I remove the cap, and then very gently, without piercing the surface of the agar, I take my needle and scrape it gently across the surface of the agar slant where the microbes are growing. I replace the cap, and now I pick up my fresh agar slant, remove the lid, and now what I want to do is take the needle and put it all the way to the bottom of the slant, and then very gently, once again, without piercing the surface of the agar, I want to skate the needle across the surface of the slant in a zigzag fashion all the way up to the top. Once again, we need to re-flame the needle in order to sterilize it. The last form of media that we're going to be inoculating is the Agar deep tube. This tube also requires the needle. And so once again, we are going to pick up a sample of inoculum from our stock tube, being careful not to pierce the surface of the agar. And now with these agar deep tubes, our goal is to take the needle and plunge it about 2/3 to 3/4 of the way into the tube in a straight line and draw out along that same line. So now we've completed our inoculation of the 3 new media from the Slant stock culture. Next, we're going to inoculate those same 3 types of media, only from the broth culture. I'm going to start again with the broth tube. Here's our stock culture. I am going to dip my loop into the stock culture, agitate it to mix the microbes, and then pull out 1 loopful of the stock culture. And if you look closely, you would be able to see how there's a sheen over the loop, kind of like on a bubble wand after you've picked up some bubbles. To transfer this into our new broth culture, we plunge it into the new broth and we twirl to dislodge the microbes. And then of course we re-sterilize our loop. Next, we're going to be using the needle to inoculate the Agar Slant and the Agar Deep. So we flame the needle. Allow it to cool. We dip the needle into our broth stock tube to coat it with microbes. And then, to transfer those microbes to the slant, we once again place the needle all the way to the bottom edge of the slant and gently spread it over the surface of the slant in a zigzag fashion up to the top. After sterilizing the needle again and allowing it to cool, we have one final inoculation to complete, and that is this final Agar deep tube. We use the needle to pick up a sample of the broth and then we take that needle and we plunge it 2/3 to 3/4 of the way into the deep tube. After a final sterilization, the inoculations are complete. The stock cultures will be disposed of, and all 6 of these tubes will be incubated at 25 degrees Celsius for 24 to 48 hours before collecting the results.